So again, welcome. Welcome to Arizona. We brought in this wonderful weather just for you. We did break 100 last week or 2 weeks ago, but we reversed that just for you guys. So welcome. So our first talk is Ramona Abbattista. Immune metabolic biosignatures associated with pathogenic mechanisms of Coccioides infection and restoration during recovery phase. Take it away.
Hello. Okay, so good morning, everyone. I'm Ramona from UC Davis, and today I'm glad to share our results on the metabolic biosignature associated with coccidiosis infection and restoration during a recovery phase. So, as you well know, coccidiosis infection can lead to a wide spectrum of disease severity, and still the mechanism of the infection, disease progression and resolution need to be established. The key answer to this question might be found in the host response, and for this reason, the hypothesis of our project is if coccidiosis infection can actually induce metabolic changes reflecting the major dysregulated host cellular pathway, which can help with the prediction of disease severity and clinical outcomes. In this context, the metabolomics can be very helpful for the determination of molecular mechanism of disease, identify biomarkers, as also pave the way to the discovery of new therapeutic targets. So in order to achieve this goal, we first want to determine the changing in the metabolic profile and functional pathways during acute phase of coccidiosis infection using serum sample. Then we target disseminated coccidiosis infection with meningitis in cerebrospinal fluid sample and serum sample. We validated then the metabolic biosignature identified for cocci infection and resolution in a longitudinal study in the context of clinical outcomes. And also we wanted to assess the potential effect of antifungal treatment on the metabolic biosignature. So the metabolomic analysis were successful to capture a massive metabolic reprogramming in the host that was revealed in the serum sample. And as you can see, the cluster analysis revealed a very good clustering between the infected group and healthy control group. And the majority of these regulated features were actually lipids, in particular fatty acid. And as you can see, there was a significant accumulation of the carboxylic fatty acid, acyl-carnitine dysregulation, which is a clear molecular sign of enhanced lipid catabolism and fatty acid oxidation. Enrichment pathway was able not only to reveal enhanced lipid catabolism, but also enhanced carbohydrate catabolism, especially enhanced glycolysis, TCA cycle, and in general metabolism for energy production, pointing towards the fact that the cell during the infection is in need of more energy, is in a condition of energy starvation. And this is typical of a condition of cellular hypoxia stress. As you can see, the levels of lactic acid were increasing across the three stages of the infection that we identified. And this is a sign of ATP dysfunction generation through the mitochondrial oxidative phosphorylation. And this regulation of energy metabolism were even more exacerbating during the disseminated stage of the infection. And indeed, we saw a drop in glucose, fructose, and carbohydrates in a cerebral spinal fluid sample. And together also with significant accumulation of medium and short chain acyl-carnitine indicating mitochondrial beta oxidation disorder. So overall, the metabolic biosignature was pointing towards mitochondrial stress and energy disbalance, which actually correlate with the main clinical feature of coccidiosis infection, which is fatigue, and very likely associated with cellular hypoxia stress. In our hypothesis, the infection could actually contribute to cellular hypoxia stress by enhancing the energy request, which is typical of infectious diseases, but also through the dysregulation of heme metabolism by the downregulation of heme. And cellular hypoxia stress can actually lead to enhanced catabolism because of energy starvation and to feed the TCA cycle and more oxygen consumption, creating this loop of cellular hypoxia stress and energy starvation where the demand of oxygen in the cell can exceed the oxygen availability. And interestingly, literature showed that cellular hypoxia stress can impair the mitochondrial function in several tissues and organs, including also liver. And today I would like to focus on the dysfunction of hepatocyte, as we saw significant regulation of bile acid metabolism, both primary and secondary bile acid, and a decrease in bile acid can be deleterious for human health, especially for the malabsorption of nutrients such as vitamins. We saw significant decrease in vitamin A and vitamin B, and actually vitamin A can be important in the context of fungal infection, as literature showed that retinoic acid and other provitamin A metabolites, they exerted antifungal activity. So this is suggesting the hypothesis that actual liver dysfunction could drive the worst outcome of the infection. The omic analysis also captured a delayed restoration of mitochondrial function in recovered patients. Indeed, we still saw a dysregulation of acylcarnitine metabolism, energy metabolism. As you can see, levels of lactic acid were still significant high in the group of recovered patients, as also dysregulation of heme metabolism with accumulation of byproducts of heme degradation, is also pro-inflammatory and antioxidant pathway. But actually what also caught our attention was the dysregulation of androsten steroid. So we wanted to understand what is the role of this steroid, and these are peculiar steroids secreted by the adrenal glands. So similarly to the case of liver, cellular hypoxia stress can induce mitochondrial dysfunction also in the adrenal glands and alter the steroid metabolism. In particular, can contribute to the shift between the so-called classic steroidogenesis pathway to a backdoor steroidogenesis pathway, which lead to the decrease of the hydroepiandrosterone, androstenediol, and corticosteroid. And the literature showed that actually low levels of the hydroepiandrosterone can affect the mitochondrial function, energy disbalance, and fatty acid oxidation, as also like involvement of adrenal gland dysfunction, alteration, and low levels of dihydroepiandrosterone were found in other systemic mycosis diseases like paracoccidiomycosis, and also cases in disseminated coccidiomycosis. And when we compare recovered patients versus diseased patients, we could actually see an increase in the dihydroepiandrosterone and its derivative, part of the classic pathway, while during the disease there was the upregulation of 5-alpha-androstenediols, which are typical of the backdoor pathway of steroidogenesis. So reinforcing the idea that actually this steroid, they could support mitochondrial function during the recovery. We evaluated the candidate metabolic biosignature in the context of a longitudinal study. So this is a case of a patient with valley fever disseminated meningitis showing a poor clinical outcome across the free visit. As you can see in the heat map, we could see a decrease in this androsten steroid together also with acylcarnitine, in particular medium short chain acylcarnitine and carnitine per se. This is another interesting case of another patient with 5 visits under a olorofim antifungal treatment, and the patient actually stopped his treatment right before the visit 4. So around the visit 4, there was a decline of the health status of the patient, and as you can see in correspondence of the visit 4, there was a spike in the secretion of the 5-alpha-androstenediol, part of the backdoor pathway of the steroidogenesis associated with the disease group. The candidate metabolic biosignature was actually independent by the antifungal treatment. In these bar plots are represented the trends of the fold changes considering the abundances variation of the selected marker compared to the control. And the trends were generally the same but between the infected group with antifungal treatment and the infected group without antifungal treatment. Although there were few exceptions, in the case of some of these androstenediol, the hydropiandrosterone, and also the bile acid, actually they were lower in the infected group without antifungal treatment. And in the case of the steroid oxy pathway, actually it was enhanced in the valley fever group with antifungal treatment. So the candidate metabolic biosignature was mainly composed by lipids and metabolism from the energy metabolism and carbohydrate and cofactor. And as you can see by the heat map and clustering analysis, it was pretty successful in the clustering of infected group versus healthy control, and also healthy group versus recovered. And also was pretty successful in distinguishing the dissemination state. So we also tested the candidate metabolic biosignature in the context of other respiratory diseases. This is the case of valley fever versus COVID. The panel was successful in distinguishing actually valley fever infected patients versus COVID patients. And interestingly, we can see that the patients with co-infection, they tend to group together in the second heat map. They tend to group together with the valley fever infected patients. What does it mean? It means that the coccidioides metabolic biosignature can dominate over the metabolic landscape of a viral disease. So the metabolomic analysis, in conclusion, were actually successful to capture this metabolic reprogramming in the cell of the host associated with energy disbalance and mitochondrial dysfunction. And mitochondrial dysfunction is actually the culprit of the dysregulation of several tissues and organs. We focus our attention today on the liver and dysregulation of adrenal glands, and which could actually could drive the process of restoration. So as a future plan, we would like to validate this panel in another valley fever patients cohort, identify potential treatment to target mitochondrial dysfunction and the correlation of cytokine expression with the trends of metabolic marker. So I would like to thank all our group, Dr. Dandekar, George Thompson, and obviously everybody for the attention. Thank you.
So we'll take just one question for this session. Kind of went a little bit over. We need to, speakers, please try to remember to keep your talk to 10 minutes. Yes.
I don't have a question. I don't think I could have one, but that's amazing.
Thank you. Thank you. Thank you very much.
Diabetes is a common risk factor for severity of disease. Did you have to break? I don't know how many patients you actually looked at here, but was there any interference from that phenotype that you had to separate out?
Yeah. So actually, thank you for the question. I was expecting this. So in our cohort of patients, we had comorbidities like hypertension and diabetes, although they were around like 12%, 15% of the total patient cohort. And I actually, so far, I didn't see cluster of sample patients within the infected patients that can be related to other comorbidities. So it seems that the factor of the disease is stronger than other factors. But as a next step, I'm gonna dig more about especially diabetes, which is a metabolic disorder connected eventually with mitochondrial stress. But so far, we didn't see internal clusters within the valley fever group. Yeah.
Next speaker, GR. Paradoxical reactions to oloriform therapy in patients undergoing treatment.